points in their life, different life phases, including weaning, commingling at a sale barn, transportation to a feed yard, or receiving at that feed yard. When these animals are stressed out, their immune systems are suppressed and they can get sick, and that illness has negative effects on their production capabilities, including reduction in their average daily gain, decreased feed efficiency, and in extreme cases it can lead to mortality. When animals arrive at a feed yard, they're typically preemptively treated with antibiotics, but there's growing consumer demand for a reduction in the dependency on these pharmacologicals, leading producers to have to find more natural ways to treat these animals or supplement these animals to keep them healthier and keep them stronger. A little bit about the physiology of stress. A stressor will stimulate the hypothalamus of the brain to secrete corticotropin-releasing hormone, which then will stimulate the pituitary gland of the brain to release adrenocorticotropin hormone, which will then stimulate the adrenal glands on the kidneys to secrete glucocorticoids and catecholamines. Some of the glucocorticoids involve things like cortisol, and those can have negative impacts on the immune cells themselves by suppressing some of their proliferation and increasing apoptosis or cellular death. Some of the catecholamines that you may be familiar with are epinephrine, norepinephrine, dopamine, things like that. And those can actually increase immune cells' susceptibility to a viral invasion, and it can also increase the pro-inflammatory response within these immune cells. The immune system is divided into two distinct but interwoven regions, including the innate and the acquired immune system. The innate immune system is, as the name implies, is what you're born with. It includes things like physical barriers, a complement system of cellular enhancement, and some of the white blood cells that I'll be talking about involved in the innate immune system include monocytes, neutrophils, basophils, and eosinophils. In the acquired immune system, these involve the humoral portion and the cell-mediated portion. In the humoral portion, we have the B cells or B lymphocytes, and these cells are responsible for producing antibodies or immunoglobulins specific to different pathogens. So this is the basis of how vaccines work. A non-toxic or a non-lethal dosage of a virus is introduced to the animal. Its acquired immune system goes to work developing these specific immunoglobulins so that in the future, if it's presented with that virus in the future, it knows how to attack it and eradicate it from the body. In the cell-mediated portion, or the intracellular portion of the acquired immune system, this involves the T cells or T lymphocytes. These cells can act as effector cells, helper cells, cytotoxic cells, or memory cells. And they're more intracellular in that they are fighting a pathogen's attempted invasion in a host cell. One of the biological responses to an immune response is the secretion of cytokines. Cytokines are chemical signaling proteins. They can be either anti-inflammatory or pro-inflammatory in their biological response within the body. Some of the anti-inflammatory cytokines that I'll be talking about include interleukin-10 or IL-10, and some of the pro-inflammatory ones include interleukin-1 or interleukin-6 or IL-1 and IL-6. Some of the common pro-inflammatory biological responses include a fever, a decrease in appetite, secretion of the adrenocorticotropin hormone, acute phase protein synthesis by the liver, and muscle catabolism. This muscle catabolism happens in an effort to mobilize amino acids from the muscle protein. It does this as the immune system is under attack. It will mobilize this amino acid in an effort to synthesize these acute phase proteins within the system and for cellular uptake by the immune cells, tissue cells, whether it's for energy or for those cells' own building blocks. They need amino acids. But we see that with this increase in acute phase protein synthesis, there's a decrease in the concentration of amino acids in the blood of sick animals. Kolditz in 2002 stated that there's a metabolic cost of disease. This is experienced when these animals are sick, they don't want to eat, which limits their growth capabilities. It also results in muscle catabolism and a loss of nitrogen retention. And this loss of nitrogen retention is a big deal because that nitrogen represents protein, and protein is expensive. That's one of the most expensive components for a feed. And so anytime there's a lack of growth or an excretion of that protein, that costs money for these producers. So it stands to reason that if you were to feed more protein, maybe it might help mitigate some of these negative issues involved with this muscle catabolism. Wagner and others in 2009 fed one group of animals at 16% crude protein and another group at 14.5% crude protein. And they found that and then they infected the animals with an endotoxin called lipopolysaccharide, or LPS. And they found that those animals that were fed at 16% crude protein had increased body temperatures. So this indicates that that excess protein, that supplemental protein, is supporting the immune cells that have a more pro-inflammatory cytokine response. Wagner and others also were investigating if they could improve the nitrogen retention of these animals by supplementing that excess protein. And while there was still a reduction in the nitrogen retention amongst those animals after that LPS infection, the animals that were supplemented at 16% crude protein had a less severe reduction in their nitrogen retention decrease. In a study by McNeil and others, sheep were administered sterile saline, indicated by the blue bars, that lipopolysaccharide, or LPS, which is indicated in the orange bars, or they were supplemented with these 6 amino acids and then they were exposed to that LPS endotoxin, and that's represented in the green bars. What they found is that after infection with LPS, there was a reduction in concentration of all the amino acids circulating in the blood of these animals. But they found that with supplementation with these 6 amino acids, there was either less severe reduction in these amino acids, or it mitigated the reduction of those amino acids altogether. Hoskin and others in 2016 executed a similar project and found similar results. And they concluded that when an immune response shifts its demand for just a single amino acid, it'll mobilize that protein, or it'll catabolize that muscle protein to mobilize for that amino acid, but in so doing is mobilizing a lot more of those amino acids, which can either be rapidly taken up by various tissue cells, immune cells, or it'll be excreted in the waste, and that'll be reflected in that loss of nitrogen retention. But it stands to reason that this supplementation with amino acids seems that it can be effective. It's just a matter of targeting with these specific amino acids. Like I stated, LPS acts as a stressor, leading to that downward cascade and increasing the levels of cortisol and the catecholamines. And this increase in glucocorticoid and catecholamine secretion can cause a variety of different biological responses, including an increase in body temperature, cortisol levels, and a decrease in the muscle mass and nitrogen retention. In a study by Nolte and others in 2008, they supplemented lambs with the branched chain amino acids, trying to target the branched chain amino acids for this immune response, and they found that that helped with the nitrogen retention of these lambs. Likewise, Leucine and others in 2018 supplemented steers with the branched chain amino acids and then exposed them to the LPS endotoxin and found that it helped the nitrogen retention reduction among those steers. So this leads us into the branched chain amino acids. The branched chain amino acids include leucine, valine, and isoleucine. They make up 35% of the amino acid total requirement. 50% of dietary branched chain amino acids bypass degradation in the liver and are metabolized in muscle, where it makes them immediately available for the synthesis of muscle proteins. The branched chain amino acids have also been implicated in insulin sensitivity, and they serve as building blocks for digestive enzymes, which implies that they may help in increasing feed efficiency or meeting those energy demands within an immune response. The branched-chain amino acids have been implicated in human, non-ruminant, and ruminant animal health. They've been seen to increase muscle protein synthesis in all these species. Petro and Bhattacharjee in 1981 found that the branched-chain amino acids were essential for the lymphocyte proliferation implying that they may be supportive for those immune cells within an immune response. Interestingly, Glassy and Furlong in 1981 found that the immune cells had a more rapid uptake of leucine versus valine and isoleucine, implying that leucine specifically may be able to supplement and support the immune response. In a study by Dardavit and others in 2002 and Wayne and others in 2017, they found that leucine supplementation led to increased muscle protein synthesis and increased muscle fiber diameter. Wayne and others in 2017 also found that leucine supplementation helped decrease protein degradation in vitro. Jose and Good in 73 found that leucine was absolutely required in vitro for the proliferation of bovine lymphocytes. Pedroso and others in 2015 found that leucine helps cells increase their uptake of glucose, implying that it can help with some of the glucose hypoglycemia often seen in LPS research. And Sands and others in 2006 found that leucine supplementation helped increase the production of some of the pancreatic digestive enzymes, which implies that it might be able to help support the immune system in making the animal more efficient with breaking down and acquiring nutrients from its feedstuffs. How leucine initiates protein synthesis is through stimulation of the mammalian target of rapamycin complex 1, or mTORC1. When leucine is available in the blood, a cell will take it in, and its presence in the cytosol of a cell stimulates the mTOR catalytic kinase to aggregate with the RAG and raptor proteins at the lysosomal membrane. Also at the lysosomal membrane are the red proteins, and when they associate with the mTORC1 complex is officially activated. The mTORC1 has been attributed to a variety of biological responses, including synthesis of protein and lipids, mitochondrial metabolism, and cellular proliferation, growth, and survival. One of the actions of mTORC1 within cells is its activation of the 4E-BP1 protein, which then dissociates from the EIF4E protein and stimulates elongation during protein translation. Along with the presence of leucine, when cytokines bind to cell receptors, it can stimulate the mTORC1 to produce the SGK1 protein, which inhibits apoptosis or cellular death within cells. mTORC1 is also associated with suppression of the ULK1 protein, which decreases muscle breakdown. Cortisol and mTORC1 appear to undergo crosstalk in that mTORC1 can suppress the transcription and translation of cortisol receptors, but likewise cortisol in the system will initiate the activation of the branched-chain amino acid transferase enzyme, which converts leucine into glutamate. It does this as a means of regulating lymphocyte proliferation to ensure that there isn't an overproduction of these lymphocytes, and so it converts that leucine and makes it less available for those cells to take in to activate that mTORC1. Within immune cells specifically, mTORC1 has been associated with the proliferation and development of both the T and B cells, and within neutrophils is responsible for the stimulation and secretion of prostaglandin, increasing vasodilation. It's also associated with monocyte recruitment to clear out infection, and binding of the IL-6 receptors to limit a pro-inflammatory response. It's also associated in neutrophils to and be involved in the transcription of the IL-10 cytokine, or the interleukin 10, and that one, as you may recall, is one of the anti-inflammatory cytokines. But none of these things happen unless there's a ample amount of leucine available to the animal to bring it into its cells. And so we thought that leucine supplementation may be one of the key steps in initiating or supporting this immune response within animals. A lot of the research that I've discussed as it pertains to ruminant animals, whether it's the cattle, sheep, or goat studies, a lot of them have been in vitro or in the lab, or they've been supplementation infused directly into the stomach or the small intestine of the animal, bypassing the rumen. This is because when an animal is directly fed leucine, the rumen microbes break it down so the animal doesn't get the same benefits of that leucine supplementation. So I developed a ruminally protected leucine supplement that we could then feed to ruminant animals. My hypothesis was that leucine supplementation would improve the immune response of lambs exposed to that lipopolysaccharide endotoxin. We chose a sheep model because they're smaller ruminants, so they're easier for me to handle, but also we could have more of them so that we would have more animals within each treatment group. The objectives of my study were to evaluate the benefits of leucine supplementation by analyzing the inflammatory and health responses, the amino acid concentrations in the blood, diet digestibility, and nitrogen retention and nitrogen balance parameters. The New Mexico State University Institutional Animal Care and Use Committee approved all procedures herein. 29 Wether lambs arrived from the Corona Range and Livestock Research Center. They were blocked by body weight into a randomized complete block design, set up as a 3x2 factorial of leucine supplementation and LPS exposure. The leucine supplementation involved an RP group, or rumen protected group. They're the animals that received that supplement that I developed. The INJ group, or injection group, received an intravenous injection of a leucine supplementation that I created. And then this control group received no leucine supplementation. Leucine was supplemented at 15 milligrams per kilogram of body weight. The animals in the injection group and control group both received a supplement as well that was of similar structure to that leucine supplementation. It just did not include that leucine. And likewise, the animals in the rumen protected and the control group also received injection with sterile saline to account for any of the fluid administration discrepancies between treatment groups. On the first day of collections, animals were given a subcutaneous injection of sterile saline that did or did not include 3 micrograms per kilogram of body weight of that LPS endotoxin. This is a timeline of my study, and it's all relative to the LPS injection. 2 weeks before LPS exposure, the animals were moved into the barn for adaptation to the environment and to get used to being handled by me. On day 7, or on day -7, 7 days before the LPS exposure, the animals were outfitted with an indwelling jugular catheter. That's how we administered that leucine solution, or the sterile saline solution, and that's when adaptation to the supplements began. 3 days before LPS exposure, the animals were moved into the individual metabolism crates to get used to that environment. Day 0 indicates the LPS injection, and on that day we also administered an ovalbumin inoculation. Ovalbumin is a protein derived from chicken egg white, something that these animals have never been exposed to. And so in theory, their acquired immune system is activated to produce immunoglobulins specific to that inoculation. It's a way that we were able to measure and test the acquired immunocompetency. After the 5-day collection period, the animals were removed from the crates. And then on day 14, we took another blood sample and gave them a booster shot of that ovalbumin inoculation. And then on day 21, that was when we did the final blood collection, and the animals were officially off study. So rectal temperatures and blood samples were taken directly before LPS exposure, and then again at hours 1, 2, 4, 8, 12, and 24 hours after LPS injection. At hours 2 and 4, we collected whole blood samples. We placed them on ice and shipped them to the Veterinary Diagnostic Services in Albuquerque for complete blood counts. At 6 p.m. on the first day of collections, we outfitted the metabolism crates with fecal collection pans and urine collection buckets, and we collected fecal samples, feed refusal urine samples, and hay and supplement samples every day for that 5-day collection period. The blood samples were analyzed for cortisol, haptoglobin, ovalbumin-specific immunoglobulins, amino acids, and glucose. The complete blood counts of the whole blood, those included all the white blood cells, the lymphocytes, eosinophils, basophils, neutrophils, and monocytes. The fecal samples, the feed refusals, and the diet samples were analyzed for all of these different components here, and the urine samples were analyzed for total nitrogen content. All data sets were analyzed using a mixed procedures of SAS. This was a randomized complete block design in a 3x2 factorial, and the experimental unit was sheep. For data sets without repeated measures, the fixed effects were LPS, leucine, and an LPS by leucine interaction. And for data sets with repeated measures, the fixed effects were LPS, leucine, LPS by leucine, LPS by hour, leucine by hour, and an LPS by leucine by hour interaction. Results were considered significant when the p-value was less than or equal to 0.05, and tendencies were considered when the p-value was less than or equal to 0.10. So what happened? No LPS by leucine by hour interactions were observed for any of the response variables. Some LPS by leucine interactions did occur, and I'll present those, as well as effects of LPS and leucine separately. Allow me to orient you to this graph. In the blue bars, we have the animals that were not exposed to LPS, and in the red bars, we have the animals that were exposed to LPS. On the x-axis, we have the different leucine treatments. So we have the control group animals, injection group, and rumen-protected group. And then on the y-axis, we have the response variable of interest. In this case, it's concentration of lymphocytes. As you can see, exposure to endotoxin caused these animals within the control group to have a lower concentration of lymphocytes compared to animals in the control group that were not exposed to LPS. There was no effect of LPS on the injection group animals or on the rumen-protected group animals. This implies that leucine supplementation was able to meet some of the demand for the proliferation of these lymphocytes and supporting those immune cells in their growth. There was a tendency for LPS to cause an increase in rectal temperatures compared to non-infected animals in the control group, but there's a tendency for no LPS effect within animals within the treatment groups of the injection group or the rumen-protected group animals. This implies that the leucine supplementation may have been ample in stimulating the mTORC1 within monocytes to secrete some of that interleukin 10 or that anti-inflammatory cytokine to help temper some of that febrile response typically seen with LPS exposure. LPS had no effect on glutamate concentration among control animals or rumen-protected animals, but LPS LPS tended to increase concentration of glutamate compared to non-infected animals among the injection group animals. This is possibly due to the activation of that branched chain amino acid transferase enzyme, converting that leucine to glutamate. This, as you may recall, the injection group animals, they got dosed with leucine directly into the bloodstream, making it rapidly available. And so to modulate that activation of the branched chain amino acid transferase enzyme may have been stimulated to kind of temper that, which would cause an increase in the glutamate concentration. LPS had no effect on glycine concentration among control group animals or rumen-protected group animals, but glycine concentration among LPS-infected animals in the injection group was a lower concentration than that of non-infected animals. Glycine is known to be associated with anti-inflammatory and antioxidative properties within the body, and so it's possible it's being utilized to help regulate some of those issues with infection. Also, in a study by Ham and others in 2016, they found that animals that were supplemented with both glycine and leucine, that those animals had a stronger metabolic response in that their muscle protein synthesis was increased. So it's possible that this glycine was taken up along with leucine in that injection group to stimulate a faster muscle protein synthesis reaction. Allow me to orient you to this graph. The blue dotted line indicates animals that were not exposed to LPS, and the red solid line indicates animals that were exposed to LPS. On the x-axis, we have time in hours relative to that LPS injection. And on the y-axis, we have the response variable of interest. In this case, it's glucose concentration in the blood. LPS caused a decrease in blood glucose at hours 8 and 12 among animals exposed to that LPS endotoxin. And typically in LPS research, after exposure to the endotoxin, there's usually a spike in blood glucose levels as the liver undergoes gluconeogenesis to meet the energy demands of that immune response. That initial spike is followed by a period of hypoglycemia as the energy demand is still high and it's, the liver's not able to undergo gluconeogenesis fast enough to keep up. So this period of hypoglycemia that we saw in our research is consistent with that. Lehman and Baum in 2004 found that leucine supplementation helped cells take glucose in at a better rate, implying that it helps with that glucose homeostasis issues. But despite this relationship, we saw no effect of leucine treatment on glucose concentration in the blood. For animals exposed to an endotoxin, circulating cortisol concentrations were higher at hour 2, 4, and 8 compared to animals not exposed to that endotoxin. This increase in blood cortisol and rectal temperature is consistent with research by Steger and others in 1999 and implies that our LPS model of stress was effective. Cortisol is also known to stimulate that branched-chain amino acid transferase enzyme, which converts that leucine to glutamate, and this increase in cortisol may also be playing a role in that elevated glutamate level that we saw among those injection group animals. LPS, as I've shown you in past research, LPS has a negative effect on blood amino acid concentrations, causing a decrease in concentration, and we saw a statistically significant decrease in leucine, isoleucine, phenylalanine, proline, and tyrosine concentrations. But as you can see, there was numerical decreases in a lot of the other amino acids as well. This is reflective of that shift that's happening within an immune response when the demand for a single amino acid changes, that muscle protein catabolism occurs to initiate that mobilization of these amino acids. And this is happening at a higher rate than these cells can be liberated, and that's what's causing that decrease. You would think that with that muscle protein catabolism, that this mobilization of amino acids would actually cause them to be increased levels because they're free-flowing, but they're being rapidly utilized, oxidized, or excreted, and that's why it's indicating that decrease in concentrations. With regard to the branched-chain amino acids specifically, we saw that LPS caused a decrease in concentration of valine at hours 4, 8, and 12, and a decrease in leucine and isoleucine concentrations at 4, 8, 12, and 24 hours after that endotoxin exposure compared to non-infected animals. This is consistent with past research in our lab as well. It may be a result of the demand by immune cells for the branched chain amino acids specifically for their proliferation may have caused that decrease in concentration in the blood, like it's being taken up so rapidly by these immune cells. Also, again, that cortisol stimulating the branched chain amino acid transferase enzyme, these branched chains may be being converted or broken down by that branched chain amino acid transferase enzyme at a higher rate. LPS infection caused a decrease in concentration for proline at hours 4, 8, 12, and 24 after LPS infection. Proline is another amino acid of interest among researchers because it's associated with muscle synthesis and weight gain in livestock animals. And so it's possible that this decrease in concentration is a result of these muscle cells taking it in faster in a period of recovery after this LPS infection. Ornithine is a metabolite of arginine, and the amino acid analysis system that I use doesn't measure for arginine, so we can use ornithine as a way of seeing the shift in arginine levels. And at hours 12, LPS caused a reduction in ornithine levels among the animals. Decreases in arginine is typically seen in LPS research. And arginine can be converted into ornithine, which can then also be converted into glutamate. And so this decrease in ornithine may also be a reason why we saw that elevated concentration of glutamate among the LPS infected animals. Allow me to orient you to this graph. In the blue bar, we have our animals that were not infected with LPS. And in the red bar, we have the animals that were infected with LPS. And on the y-axis, we have total white blood cell concentration. As you can see, LPS caused a decrease in that total white blood cell concentration, which is consistent with research by Blecka and others in 1984, where stressed and immunosuppressed animals had a decrease in their white blood cell counts. We saw a numerical decrease in eosinophils, basophils, monocytes, and neutrophils, but the only statistically significant decrease was that of the total white blood cells and the lymphocytes. 24 hours after LPS exposure, circulating concentrations of haptoglobin were elevated for the LPS-exposed animals. Haptoglobin is one of those acute phase proteins that I mentioned earlier that's synthesized by the liver. They are associated with anti-inflammatory properties. They're also known to have higher concentrations of phenylalanine, lysine, serine, tryptophan, and cysteine. And this may be a cause of why we saw a statistically different concentration of phenylalanine, lysine, and serine in the blood of these sheep, was that those amino acids were being mobilized to the liver for the synthesis of haptoglobin. One of the actions of haptoglobin is it binds free hemoglobin, and hemoglobin carries iron, and pathogens use that iron for their own cellular growth. So by binding it up, haptoglobin is reducing the ability of those pathogens to undergo their own proliferation within the body. We saw no treatment effects on the concentrations of the ovalbumin-specific immunoglobulins. Some of the past research that I mentioned had stated that the branched chain amino acid supplementation enhances the vaccine response, and so we had hoped that the leucine supplementation would increase the concentration of the ovalbumin-specific immunoglobulins within these animals, but unfortunately that wasn't the case. This may have been because we supplemented too little leucine, or because these cells may have a requirement for a balance of all three of those branched chain amino acids. Allow me to orient you to this graph. The green solid line indicates the animals on the control diet. The orange dotted line indicates the injection group animals, and the purple dotted line indicates that rumen-protected supplement group. On the x-axis, we have time in hours relative to LPS injection, and on the y-axis, we have leucine concentration. Leucine concentration was the only response variable that had an LPS by hour effect. At hour 0, the injection group animals had higher levels of leucine in the blood than that of the control animals, and tended to have higher levels than that of the rumen-protected animals. And as you can see, the rumen-protected animals had a numerically higher concentration than the control animals. And this implies that our adaptation at mealtimes, the supplementation, was effective in getting that leucine to the bloodstream of these animals. At hour 12, the injection group and rumen-protected group animals had lower levels of leucine concentration than that of the control animals. And this is possibly a result of that available leucine being utilized for that recovery response after that LPS infection. There was no effective treatment on dry matter intake or digestibility within this study. However, with fat intake and digestibility, we saw statistically significant results in that the rumen-protected group animals had a lower intake of fat. This is likely due to the composition of the supplement themselves, though proportionally, the one that contained leucine had some leucine, whereas the control supplement was just all fat. So it was 100% of one thing versus a lower percentage. So it makes sense that their intake of fat was lower because their diet had less fat in it. Likewise, their fecal content was also lower in fat. However, the decrease in the fecal content of fat may have also been the result of some of the rumen microbes undergoing beta oxidation of the fat molecules in the rumen, which would lead to less fat in the feces. But interestingly, we saw a decrease in the digestibility. The total tract fat digestibility decreased among those rumen-protected group animals. And this actually implies that the rumen-protected supplement was successful in bypassing degradation and may have actually been protected from the rumen as it was less digestible. The ADF intake and digestibility shifted for the animals within the rumen-protected group. ADF stands for acid detergent fiber, and it's an analysis that we do to determine the plant fiber content within the feedstuffs. As you can see, there was no difference in intake, but there was a much higher tendency for a higher amount of ADF within the feces of the rumen-protected animals. And thus there is a tendency for a decrease in the ADF digestibility among those rumen-protected animals. This is again possibly due to the composition of this supplement. It's made from a variety of lipids or fats, and one of which was a less saturated fat. It's possible that fat was mobilized in the rumen, and it can coat the feedstuffs, and then the cellulolytic action of that bacteria can't break that feedstuff down, and so it just bypasses through the system undegraded. So that could possibly be why we saw that decrease in that digestibility. That may also explain the decrease we saw in the fat digestibility, the inhibition of those rumen microbes from that fat presence within the rumen itself. I want to point out that there was a numerical decrease in the digestibility of all the other nutrients among the rumen-protected animals, further implying that those bacteria may have been inhibited by some of the fat content of that supplement. I would also like to point out that there is no effective treatment with regard to the nitrogen retention. There was an effective leucine treatment on the intake, but as you can see, the higher intake is among those animals that received that rumen-protected supplement, but that makes sense because we added leucine, which has nitrogen. So we're not surprised that that one was higher, but there was no effective treatment on the nitrogen retention. So LPS typically causes that decrease in nitrogen retention, so it's interesting that we didn't see this same decrease in our research. The objectives of my study were to evaluate these benefits of leucine by analyzing the inflammatory and health responses, the blood amino acid concentrations, diet digestibility, and nitrogen retention parameters. And therefore I successfully met all of my objectives for my study. But but what exactly does it all mean? Leucine supplementation seemed to have an interesting impact on the proliferation of the lymphocytes. In it, there was no effect of LPS treatment among the animals that received that supplementation, so that is interesting. Likewise, the leucine supplementation seemed to have tempered some of that pro-inflammatory and febrile response that we typically see with LPS-infected animals. However, I think that we didn't see some reactions that we may have expected because we didn't supplement enough leucine. And so in the future, going into this, we would need to make modifications to increase the leucine content of that rumen-protected supplement, but also make modifications to the composition of that supplement itself to eliminate some of those digestibility issues that we were seeing. Likewise, I think that we need to increase the LPS dosage to see some of those nitrogen parameters that we were looking for. We dosed a subcutaneous injection of 3 micrograms per kilogram of body weight for LPS, and so I think that the study shows that it would need to be upwards of 4 micrograms in the future to stimulate those responses that we usually see with regard to nitrogen retention. But with all this in mind, I'm ready to partially accept my hypothesis because the leucine supplementation did appear to improve some of the responses within the immune system. When I was in my sophomore year at my undergrad, I took a biology class. It was my first life science classes at a college level, and I had no idea what I was doing. And after the first quiz, I got a D on the first quiz. And after she handed back the quizzes, the professor told me that I would never academically succeed in the hard sciences and that I should drop that class. And I took her advice and dropped the class, but now today I'm standing up here defending my thesis for my Master's of Science, and that would not have been possible, sorry, this is the hard part. That would not have been possible without the support of my family. They didn't doubt my abilities when I decided I wanted to go back to school for veterinary school. They were just like on board the entire time. And without my family and my friends and their support, there's no way I would have been able to do this. Oh, the Aggie Cupboard, they're the on-campus food pantry. They're awesome. Meg is here, she's the director. And right now they're doing their turkey drive for the holiday season. And for $25, you can buy a turkey for a there's gonna be a lot of beer drinking in the next few slides, warn you. You can buy a turkey, a holiday turkey for families in need in the NMSU community for just $25. So thank you to the Aggie Cupboard for everything they do. It's been a privilege to be one of their volunteers. I'd also like to thank my committee for their patience during this process. Dr. Eric Scholigurtis, Dr. Omar Holguin, and Dr. Clint Lust. They've been really helpful throughout this process. I'd also like to thank all of the professors I've had here at NMSU, some of whom are here today. I didn't know a single soul when I came here, and so I just piled everything into my classes when I came here, and I'm really grateful that I've gotten to know some of the professors that I've had in my time here. And also, I'd like to thank all of my friends and my fellow grad students who have made this experience like way more fun. Because, man, without the support of my colleagues, like, it would just be tough every single day. There'd be more tears than there was already. I'd also like to thank the WeSTEM Club. Their current president, vice president, volunteer coordinator are here. I served as the president last year, and it was like one of the highlights of my time at NMSU. And being their president just was super fun, and I got to meet a lot of really awesome people in that process as well. And yeah, I really appreciate everyone being here today. And with that, I'd be happy to answer any questions you may have.